Hello everyone and welcome back to PhytoPathoVerse. I'm Malini Ray, molecular plant pathologist with a motto to make plant pathology easy. So in our last episode, I shared with you the table of contents, okay? So that today we could start chapter 1 easily, that is the history and development of plant pathology. Interestingly, this chapter contains 57 pages, which is a lot, but not to worry. We will just break it down into clear, simplified concepts and wrap it under 1 hour. Before we start, don't forget to stay till the end because I will be sharing takeaway questions to test the understanding. So let's get started. So to start with, we have here prologue. If anybody knows, they can comment down, but I have to say it. Prologue is the introductory section, means the background check, right? We are entering into a new thing, so obviously we have to do the background check, but we will do it very fast so that we can jump into the main section of history. Okay, so here we see plant pathology is the science that studies plant diseases, like how pathogen cause disease, how plant resist them, how disease spread, and how they can be managed or controlled. Here, it integrates with many other disciplines like microbiology, biochemistry, genetics, cell biology, ecology. It also collaborates with engineering, economics, agronomy, and with science. Yeah, also entomology. See, it is not always bad. It is also a successful science. Bad in a sense, always talking about, diseases and how it can be harmful. So it is good as well if we learn how to manage the diseases caused by various pathogens. So yes, it is also beneficial. It is beneficial. So it is a successful science. Like here we can see global human expectancy, life expectancy increased from 34 years in 1913 to over 72 years in 2015. But still 17 to 30% of major crops are lost to pathogens and pests, even in developed countries. Also, there are post-harvest losses, which is approx 10 to 30%. Okay, so there are more examples, reference, case studies. You can look into that. Next, we will check this section, plant and diseases. This is better than the last one. Okay, plants make up the majority of the Earth's living environment in forest, grasslands, shrubs, as well as plantation, pastures, edible, and so on, so on. So, directly or indirectly, plants also make up the food on which humans and all animals depend. We know we have learned so much about plants in our schools, everywhere in our biology, science, everywhere. Key plants are autotrophs and they are our food source. Herbivores, omnivores, whoever it is, plant is the main source because they can prepare their own food. Okay, but same as animals and humans, plants get sick too. They also get sick. Sick plants grow and reproduce poorly. They exhibit various types of symptoms and often parts of plant or the whole plant die. Okay. The agents that cause disease in plants are the same or similar to those causing disease in humans and animals. They include pathogenic microorganisms such as virus, bacteria, molecules, fungi, oomycetes, nematodes, unfavorable conditions such as, abiotic stresses such as lack or excess of nutrients, moisture, light, presence of toxic chemicals, air, and soil. But here we need to remember one more thing. Key, external damages. External damages caused by macroscopic animals is not usually included in the study of plant pathology. Normally in plant pathology, we usually observe or study about the organism which is microscopic, right? So normally we say microscopic organism which is harmful, and it is studied under plant pathology, not the macroscopic one, not the macroscopic animals. All right. Now they have given one definition here of plant pathology. We can look. Plant pathology is the study of the organisms and the environmental factors that cause disease in plants. The mechanisms by which these factors include disease in plants, and the methods of preventing or controlling disease and reducing the damage it causes. All right. Till this clear? Again, they are saying plant pathology is for plants largely what medicine is for humans. That is correct. Humans are important, animals are important. Similarly, our plants are also important, very, very important. So, the same way medicine is for humans, veterinary medicine is for animals, the same way plant pathology is for plants. Okay. Each discipline studies the causes, mechanisms, and control of disease affecting the organism with which it deals, that is, plants, humans, and animals. Very well said. As a science, plant pathology tries to increase our knowledge about plant diseases. There are very few knowledge. Like we have so much knowledge about human disease. Obviously, we are humans, so we will have that. But plants are everywhere. In the first line, we have seen plants make up the majority of the Earth's living environment. Also, we have seen directly and indirectly plants make up the food on which humans and all animals depend. So obviously, knowing about what is harmful for plants, knowing about how to manage what is harming the plants, it's very important. Okay. Next, we will start with the concept of disease in plants. Now, the concept of disease means what? Means how to understand, how to differentiate whether a plant is healthy or diseased, right? So, it is accepted that a plant is healthy or normal when it can carry out its physiological functions to the best of its genetic potential. Physiological functions means, respiration, plant does respire, right? They have these things. Photosynthesis, don't go with my handwriting. Okay. Respiration, photosynthesis, and all the other physiological functions, when they carry out their physiological functions to the best of its genetic potential, then it is referred to as healthy plant. But when, from here we will see, when the ability of the cell of a plant or plant part to carry out one or more of these essential function is interfered with by either pathogenic organism or any adverse environmental factors. Now, what are the pathogenic organisms and the environmental factors? We will be seeing that in few minutes. So the activities of the cells are disrupted, altered, or inhibited. The cells malfunction or die, and the plant becomes diseased. Simple. When they can carry forward their physiological functions to the best of their genetic potential, they are considered as healthy. When they cannot do that, the physiological functions are interfered, So the plant is considered as diseased. Okay. Now, disease in plants then can be defined. How it can be defined? Disease in plants. It can be defined as a series of invisible and visible responses of plant cells and tissues. This much is fine. Now, as I have told, we will look into the pathogenic organism and adverse environment which harm and make a plant diseased. They are here. See, here are the names. We can also see here visually, left hand side, you can see all the healthy things. Left side, we can see normal. The plant is normal. Right hand side, we can see the plant is diseased. Okay. Basic function of a plant, see left side. And right side is interference with these functions. Fine. Okay, so the biotic factors, the pathogenic factors here are fungi, oomycetes, prokaryotes like the bacteria or molecules, parasites of higher fungi, plant parasitic higher fungi, viruses, viroids, nematodes, rhizia. These are the pathogenic um, interferences. These are living, that's why it's called biotic. Also, we have abiotic, which is non-living. That means it is environmental interferences. Means due to lack or excess of soil moisture, temperature, oxygen, light, deficiency, toxicity, alkalinity, and so on and so forth. Okay. History of plant pathology and early significance of plant diseases. Okay, there is nothing much to discuss here, only story. When humans were hunters and gatherers and all the stories, so we can skip that. In this image, although you can observe the shapes and size of certain plant pathogens, that is, see, this is the nematode head and bacteria. Yeah, everything is written here. Okay. Mycelium, how viroids or virus they looked, we can see this image for the shape and size of various plant pathogens. Also, in this we can look into the morphology and way of multiplication of some of the groups of plant pathogens. Here we can see nematodes, virus, parasitic higher plants, molecules, bacteria, fungi. Yeah, most of them are here. Okay, then all right. Biology and plant pathology in the early Renaissance. Okay, so shift from superstition to observation. Shift from superstition to observation began in this Renaissance. What we can see here. People continue to suffer from hunger and malnutrition due partially at least to diseases destroying their crops and their fruit. They, however, continue to consider plant diseases as the work and wish of their god, superstition. And therefore, an event that could neither be understood nor avoided. That's it, to understand what superstition is. But at that point of time, they were not having any answers. That's why they considered it is the wish of the god. But nowadays, we have all the answers, but normal people, generally, we don't want to know the reasons. That's why we are unaware and still uneducated about the part that there are so many causes of plant diseases and they can be controlled. And the losses can be reduced or minimalized. Okay. So there are various contributions and concepts, thoughts, discoveries of various scientists that will be looking here. In 1670, French physician Tollier observed that ergotism or holy fire, a serious and often deadly disease of humans in North-Central Europe, did not spread from one person to another but seemed to be associated with the consumption of ergot-contaminated grains. This is a very severe disease. Even today, we consider this ergotism, holy fire, one of the major historical events that we study even now. Can anybody tell me what is the pathogen that causes ergotism? This is a homework. You can try, you can search if you want to know the answer, and yeah, answer to me in the comment section. I can correct it or I'll answer it in the next episode. Okay. Then coming to this, we have, okay, before that, again in the figure, please look into the figure very nicely because the questions from NET, SRF directly comes from the images. I'll tell you one question like this, high temperature, okay? There is a question is Manoj Kumar Bhattacharya. Manoj Kumar Bhattacharya, if anybody is from agriculture, he or she will know. There is one question from MK Yadav. Sorry, Manoj Kumar Yadav, not Bhattacharya, sorry. MK Yadav is that the diseases which is caused due to high temperature, sun scald, water core, and blossom end rot. And I got to see a question while doing some mock tests. So there I have seen four options were there, and the fourth options were something. Some options were there, I don't remember. A, B, C, D. Now, everybody of us thought the answer will be this. Means, sorry, the question was which of the following is not due to high temperature? Which of the following disease is not caused due to high temperature? Okay. So most of us considered, we did not know about this fourth disease, but we were very sure blossom end rot is not the answer. It cannot be due to high temperature because normally blossom end rot, how we learn it, it is because of calcium deficiency. But it is also because of high temperature. So this is one of the questions when they give MCQs directly from the figures. Okay, so look into the figures properly. Coming to the text, in 1735, Swedish philosopher botanist Carl von Linné published his main work, Systema Naturae. Whatever I'm underlining, please note that because again, direct question comes from this key, which is the book that was written by Carl von Linné. Okay, that is Systema Naturae. Okay, so by this book, he established the systematics of plant species and the binomial nomenclature. Binomial nomenclature means whoever is having biology in 10 plus 2, he or she will understand. Binomial nomenclature means the naming of organism. They consider genus and species, okay? Binomial nomenclature, two names. Okay, linear species, however, were rigid and were supposed to have remained unchanged since creation. It was not until more than a century later, in 1859, the Charles Darwin published book The Origin of Species. This is one more important thing, written by Charles Darwin, The Origin of Species, by means of natural selection. This is the total, sorry. The Origin of Species, 1859, Charles Darwin written The Origin of Species by means of natural selection. and showed that the species of all organisms, plants and animals evolve over time and adapt to changes in their environment for survival. Okay. In 1729, Italian botanist Pier Antonio Micheli, Italian name, it's different. P.A. Micheli described many new genera of fungi and illustrated their reproductive structures. Okay. So, with this P.A. Micheli, various question comes. One is, he is considered as the father of mycology. Okay. Also, he has studied, he has worked, he has worked many uh, on mycology and the concept of mycology. We will come to that topic when we will start discussing mycology part. Not to bother you with all these things. Just remember, P.A. Micheli is considered as father of mycology. Moving on. Um, 1743, British scientist Needham observed nematodes inside small, abnormally rounded wheat kernels, but he too failed to show or suggest that they were the cause of the problem. Yeah, he failed to do so, but yes, he observed the nematodes for the first time. And the nematode name, if you can answer it in the comment box, that is fine, but I would love to say that it is Anguina treatisi. Okay, it causes seed gall in wheat kernels that we can see here. So the first nematode that was observed by Needham in 1743, that was Anguina treatisi. All right. Okay. In 1755, the Frenchman Tillet, working with smut wheat, showed that he could increase the number of wheat plants developing covered smut by dusting wheat kernels before planting with smut dust, smut spores. Fine. But can anybody again tell me by what can we call Tillet? We have heard of father of plant pathology, father of mycology, father of bacteriology, father of, botany and everything. Till now, I have heard Tillet is only called the great grandfather of plant pathology. Okay, the great grandfather of plant pathology is Tillet. That's interesting. Well, he has worked on smutted wheat. Okay. Okay. He also noted that he could reduce the number of smutted wheat plants produced by treating the smutted kernels with copper sulfate. Yeah. Okay. Okay. Tillet too, however, did not interpret his experiments properly. He has this idea, but he did not interpret his experiments properly. Instead of concluding that wheat smut is an infectious plant disease, he believed that it was a poisonous substance contained in the smut dust rather than the living spores and fungus coming from them that caused the disease. But more than 50 years later, in 1807, it was Prevost, another Frenchman, repeated both the inoculation experiments and those in which the seeds were treated with copper sulfate as done by Tillet and obtained the same results. Means, first the idea was initiated by Tillet, but for more than 50 years later, in 1807, Prevost conducted the experiments. He followed the route started by Tillett, and at last, what he did, in addition, Prevost observed that untreated and treated seeds, wheat seeds under the microscope, and noticed that smut spores from untreated seeds germinated and grew, whereas those from treated seeds failed to germinate. He therefore concluded correctly that it was the smut spores that caused the smut disease in wheat, and that the reduced number of smutted wheat plants derived from copper sulfate treated seed was due to the inhibition of germination of smut spores by copper sulfate. So whenever we will see a question, a question is asked from the part of chemicals or history, anywhere, he who first discovered or used the copper sulfate. Where is this? Copper sulfate. So although the idea was initiated by Tillett, but the answer will be Prevost. Prevost in 1807, he derived, he concluded that copper sulfate is responsible for the smut-free wheat seeds. Okay. Okay, so what more? Okay, let's see what's there. Prevost's conclusion, however, were not accepted by the French Academy. Okay, it was not accepted. Yes, any new ideas, any new concept, any new theories, whatever will come for the first time, obviously it will not be accepted because we are habituated with one kind of environment with some pre-conceptualized events that we have in our mind. So anybody is coming up with new ideas, at first it will not be accepted. Okay, again, Prevost's conclusions, however, were not accepted by the French Academy of Sciences because um, other scientists throughout Europe still believed, because the scientists still believed that microorganisms and their spores form through spontaneous generations and were the results rather than cause of the disease. In 1855, a nematode was observed in the galls of cucumber roots, but again they were thought to have appeared there spontaneously. Fine. Now, this is a box. Again, we will see lots of boxes. I'll tell what the box signifies. See, lots of boxes. So, okay, now as we can see, there are several important historical events like this one, Great Irish Famine. Okay, and concept discussed in detail within this text boxes, like wrath of gods. I think here I have seen that. See, wrath of gods. Then Irish famine, Robigalia, Turkey X disease. This is Irish famine, Turkey X disease, Chilipomania. There are further boxes that we will be seeing in the next parts. Koch postulates, Bordeaux mixture, UG99, chestnut blight, Dutch elm disease, ergotism, mycotoxins, and many more. So what I have thought is that to give each one of this the attention, the true attention they deserve, because these historical events are very, very important. So while covering this whole session, discussing about each one of this event, it will not be justified. So to give the real attention they deserve, we will be covering them separately in a fresh session. This way, we won't miss any single point. We will understand everything clearly, and we can also discuss the types of questions, the types of MCQs, the types of, um, questions that comes in competitive exams. Okay, with this, as this page is already open, here I'll say a few things, one or two things of Great Irish Mine. Because of this great Irish famine, we got to know about the first fungal disease, first fungal pathogen. Okay, so this is very important. Great Irish famine was caused due to, see, potato. It was seen in potato. So the disease name was late blight of potato, 1845, and this disease was caused by one pathogen, one fungi actually, Phytophthora infestans. Okay, this is the pathogen name which caused this late blight of potato 1845, because of which we have this great Irish famine, and it was an epidemic. There are so many terms called epidemic, endemic, sporadic, pandemic. We will cover all those in that in epidemiology chapter. Okay. Filamentous microorganisms are the cause of many plant diseases. Now, what this filamentous microorganism means thread-like. The shape should be thread-like. It is normally fungi or bacteria. Bacteria which grow in a thread-like shape. Thread-like shape. Okay. So there's nothing much to say here. See, the same things are repeated. Tillate, Prevost. Uh, let's see what's there. Um, Prevost, also in France, repeated and expanded Tillet's experiment and appeared to have demonstrated conclusively that wheat smut was caused by a fungus. But it was still widely believed that spores were the result rather than the cause of the disease. This belief continues to be shared and expounded by scientists for at least 50 years more. Okay, fine. Same old, nothing much to discuss. But in this section, one important, very important question I have seen, and I want you to learn it, like mug it up. That is this two line. The scientist's name is Kuhn, and he wrote the first ever book on plant pathology. The book name is Diseases of Cultivated Crops, Their Causes and Their Control in 1858. That's it. Just learn it. This is the important point from this section. Next, we will see the discoveries. Discoveries of causes of disease. Causes of disease means one we are seeing here nematode. Next we will see fungi, bacteria, viruses, viroids, molecules and all. Okay. Start with nematode. In the last section we have discussed the first nematode was discovered by Needham in 1743. I also told you the name, that is Angina treatisi, okay, which caused seed gall. Again, I'm saying, okay. The next is bacteria. For bacteria, the first scientist who caused diseases in plant pathogenic bacteria. The first bacterial disease was not plant pathogenic; it was human. But in plant pathogenic bacteria, the first was discovered by T.J. Burrell in fire blight of apple and pear. Okay. The pathogen which causes fire blight of apple and pear is Erwina amylovora. Okay, Erwina amylovora. Okay. Okay, bacteria. First nematode, then bacteria we can call it like second. Third we have viruses. I don't see fungi here. Maybe fungi won that Great Irish Famine. That time we have discussed the first plant pathogenic bacteria. It was lead blight of potato by Phytophthora infestans. That will be fungi. That's why it is not discussed again and again. For virus, it is interesting. We will cover it more. Okay, we will see whatever it's written. At about the same time that more diseases of plants were shown to be caused by bacteria, the Dutchman Adolf Meyer, note the names, in 1886 injected juice uh, obtained from tobacco plant leaves showing various patterns of greenish yellow mosaic. This term from all this question comes. The mosaic term was first used by whom? The scientist's name is Mayer. Mosaic Mayer, MM, remember like that. Into healthy tobacco plant, and the latter developed similar mosaic patterns. Because no fungus was present on the plant or in filtered juice, Mayer concluded that the disease was probably caused by a bacteria. Fine. Mayer's conclusion was the disease is caused by a bacteria. Later on, Ivanovsky, one more scientist, note the name as well, showed that whatever caused the tobacco mosaic disease could pass through a filter that retains bacteria. There is a bacterial filter filter. The smallest size of bacteria, even the smallest size of bacteria will be filtered out from that filter. But virus can pass through that filter, bacterial filter, right? So he concluded that the disease was caused by a toxin secreted by bacteria, or perhaps by usually small bacteria that pass through the pores of the filter, not normal bacteria. Next scientist, again continuing the work, Beijering, by repeating some of these experiments, finally concluded that the tobacco mosaic disease was caused not by a microorganism but by a contagious living fluid that he called as virus. So the question can come, the term virus, it was given by Beijering. The term mosaic was used by Mayer. Okay. Ivanovsky, what he did, he proved that the microorganism causing tobacco mosaic disease, it passes through bacterial filter. Okay, one more scientist is there, Stanley. He crystallized, because nobody knew the true nature or shape of the virus. So he crystallized the virus with ammonium sulfate, crystalline protein obtained from crystalline protein. Like Stanley added ammonium sulfate to tobacco juice extracted from infected tobacco leaves and obtained a crystalline protein that, when rubbed on tobacco, cause the tobacco mosaic disease. Simple. This four scientists' name is very important. One is Mayer, second is Ivanovsky, third is Beijering, fourth is Stanley. So who is the father of virology? Anybody knows? Father of virology. Father of virology. Okay. Also, I have one more question for you from this. Okay, I'll give it A because already one number is there. B, question B. Does anybody know who is called mother of virology? This is very interesting because, like the great grandfather of plant pathology, this is one more thing, the mother of virology. Normally, we see all the fathers everywhere. It's very rare to see, mother of virology is also there. So please go and find who is the father and mother of virology and comment me on comment the answer in the comment box. If not, I will surely answer it in my next episode. Okay. Okay. Next we have viroids. Viroid is very simple. It is very understudied, and it causes disease only in plants. Remember, viroid causes causes disease only in plants. Okay. Um, in 1971, studies of potato spindle tuber disease, uh, Dinier. Okay, in normal sense, I'll tell. The first disease in which the viral was first discovered was potato spindle tuber disease, and the scientist name was Dinier. And from here itself, many questions can come like which is the first disease, which is obviously potato spindle tuber virus. Also, more questions come like the genetic material and all everything that we will cover while studying the virology section. Also, which is the largest viroid, which is the smallest viroid. Okay. Next, molecules. Molecules, it can be phytoplasma, mycoplasma, whatever it is. It was discovered by Doi et al. Colleagues. In scientifically we write if in scientific journals, if there are more than two authors, we can write if it's Doi is the surname, it will be the surname, Doi et al. Okay. Like this, we write it down. So, Dewey et al. In 1967, he discovered the molecules. Okay, let's see a little bit about the management history, starting with chemicals. So, to know this, we have to know about Millardet, PMA Millardet in 1885. He discovered Bordeaux mixture, which is a combination of copper sulfate and hydrated lime. Bordeaux mixture is a serendipity discovery. Okay. Now, what that means? It means sudden discovery. But it was against what? It was for what? It was for downy mildew of grapes caused by Plasma viticola. Okay? Okay. So, for more than 100 years, Bordeaux mixture was used more than any other fungicide against a wide variety of plant disease all over the world. Okay, that's it. Next, we have, it's getting very lengthy. Let's see how to wrap it up. Okay, in 1928, we'll go fast now. In 1928, Alexander Fleming discovered penicillin. We know this, and it is used against bacterial disease of plants, and was considered unlikely for at least the next 20 years. Okay. Here again we have in 1934 the first dithiocarbamide fungicide, that is therum, was discovered, which led to the development of series of effective and widely used fungicide using verpam, zinab, and manab, means the sulfur fungicides. Okay, dithiocarbamides, it's nothing other than the sulfur fungicides, and it is a sulfur fungicides are normally contact fungicides. Okay. Okay. Here one more thing we have skipped is that this is important. 1965, the first systemic fungicide. Okay, somewhere you can see 1965, somewhere you can see it will be 1966, in which the first systemic fungicide, carboxyl. I have told you, right, before some time this sulfur fungicide are contact. The copper fungicide we have discussed about the Bordeaux mixture, copper sulfate and hydrated lime, that was copper fungicide, and this is the sulfur fungicide. Both are contacts. Okay, but there are also few fungicide group which are systemic in nature, out of which the first is carboxyl, which was discovered in 1966 by Vaughan and Kulka. Vaughan and Kulka discovered first systemic fungicide carboxyl 1965 or 6. Carboxyl is also called Vitavax, and it is normally used against smut diseases. Okay, okay. Uh, Next, the appearance of pathogen races resistant to antibiotic and fungicides. Okay, so there are lots of antibiotics and fungicides, but pathogens are very clever. Okay, they are also clever, and so there are various races. So if there is one type of pathogen and we have developed resistant variety against it, so they can also be resistant against the antibiotics and fungicides that we have prepared as a medicine to kill them, right? So let's see how it goes. In 1954, it was noticed that a few strains of bacteria causing disease in plants were resistant to certain antibiotics, and in 1963, strains of fungal plant pathogens were found that were resistant to certain protective fungicides. So there were resistance against antibiotics also, fungicides also. Cool. Okay, the appearance of pathogen races resistant to chemicals prompted the development of new strategies. Yes, if they are developing, they are building new strategies to means evolve into new races. So we also have, so we will also develop new strategies to make more new generation fungicides or antibiotics or different mode of action fungicides which they are not yet familiar with. Okay, okay. Now, yes, public concern about chemical pesticides. So chemical pesticides are obviously chemicals, right? The name chemical is there, so public concern will be there. So it is inevitable that chemical pesticides are toxic poisons. Yes, the word pesticide itself means pest killer. So depending on the kind of pest against which they are effective, pesticides are known as antibiotics. Like if it's bacteria, then it's called bactericides, fungi, then fungicides, nematode, then nematicides, insect, then insecticide, herbs means herbicides, and so on and so forth. Okay. Okay. In this, one more important question that you should note that the book Silent Spring was written by Rachel Carson, and it describes the danger of polluting the environment and poisonous chemicals that we're talking about the this public concern, right? Of this chemical pesticides. This is the book written by Rachel Carson, which describes the dangers of polluting the environment with poisonous chemicals and documented several cases of bird and fishes deaths to be the result of pesticides being accumulated and concentrated through the food chain. Carson's book generated a great deal of controversy, but also a much greater awareness of the possible adverse effects of pesticides. Okay. Okay. Many scientists at first were quite skeptical and unconvinced of Carson's arguments. Little by little, however, many of them agreed to do research on the issues of the safety pesticides. Okay. There were many more events, like, by the mid of 1960s, all the pesticides contain mercury, because mercury is also a contact fungicide, and because it is again that metal. So, we use very little amount of mercury, like for 0.1%, but there are various mercury chlorides, organomercurials that has been used in history, but after a point of time, by the mid 1960s, they were banned by the US and the other governments. Okay. So, okay, rather than chemicals, there are few alternatives to control plant diseases. One of which, obviously, something is coming in our mind, that is cultural method. So proper cultural practices include removal of plant debris and infected plant parts, use of seed-free pathogens, crop rotation, and plant species that are immune to the kinds of pathogens that affect the other crop rotation, soil fallows, and so on. Okay, that means cultural practices. Other than cultural practices, we have biological control. Okay, plant diseases with microorganism antagonistic to the pathogen has emerged to become a major part of the crop protection. Biological control, we normally do it against soil-borne pathogen, like some agents are like trichoderma, pseudomonas, and all those. Okay. Okay, we have almost come to an end of this session. Only little bit is left. Okay, this is epidemiology of plant disease. Epidemiology is again very important because if we have heard about the disease triangle, then three things is important, that is susceptible host, virulent pathogen, and favorable environment. Environment means epidemiology, that's it. And the father of epidemiology is Bo van der Planck. So in 1963, van der Planck wrote this book, Plant Disease Epidemics and Control, established epidemiology as an important and interesting field in plant pathology. Okay. One more thing that comes in exam is that 1963, which one is the first computer simulation model and against what disease? There are various computer simulation models which help us understand or focus a disease which is about to come. The first one is APDEM, okay, against early blight disease of tomato and potato. That's it. Heading towards molecular plant pathology. Since 1980, great emphasis has been placed on determining the specific molecules involved in disease development. This reductionist approach became known as molecular plant pathology. One more homework for you. Yeah, I'm giving lots of homeworks, but I don't know. Can you tell me who is again the father of molecular plant pathology? Okay, father of molecular plant pathology. Simple. Everywhere I'm just asking about the father and mother. Same, father of molecular plant pathology. Okay. The major step forward became when Beadle and Tatum used mutants of Neurospora tracer in 1941 to show that one gene codes for one enzyme. Okay, Neurospora is a genetic plant. It is used for genetic studies. It is a laboratory plant which is normally used for genetic studies. Okay. Then what we can see here. Um, by the mid of 1970s, studies of agrobacterium tumefaciens revealed that the ddNA of its TI plasmid contains several genes, of which two coding or two regulators were responsible for production of tumor galls. Okay, okay, okay. Interesting things are coming. Interesting things, and also important for exam. Yeah, just got it. This is the para. This is the very, very important paragraph. Okay. Discoveries in molecular plant pathology accelerated in 1990s. The concept of systemic acquired resistance burst into the scene through the discovery of uh, Craig's and colleagues and Ryals and co-workers that salicylic acid is relative of aspirin is associated with ACR. The first fungal a virulent gene, aviar, was isolated from Cladosporium fulvum by DeWitt, while the first plant-resistant gene HM1 was isolated from corn by Stephen Briggs and Jonathan Walton. The latter also showed that HM1 operates by producing a protein that detoxifies the host selective toxin of the pathogen called Cochliobolus carborum. Okay, what should we study from here? The first thing is first fungal avirulent gene. Okay, this was the first thing that we should learn. That is AVR9 by DeWitt. Okay, and from Cladosporium fulvum. The second thing that we should know from this point is that portion is that first R gene, that is the resistant gene, is HM1 isolated from maize by whom? By Briggs and Walton. Okay, please note it down. The first fungal aviary gene is AVR9 by DeWitt from Cladosporium fulvum, and first resistant gene HM1 in maize isolated from corn or maize by Briggs and Walton. That's it. Okay. Advances, let's see about the advances. Advances in molecular plant pathology have also provided a new set of diagnostic tools and techniques that are used to detect and identify pathogens even when they are present. Yeah, there are so many diagnostic things that they have discussed. There are so many, like CRISPR-Cas has come, NGS, next generation sequencing, Sanger sequencing, and many more. Mostly they nowadays they are using this application of genomics. Genomics means genes, the use of genes. Even for the classification of fungi, they are using for on the basis of genes. So yeah, from nowadays advances have been maintained by the application of genes. Okay. To exploit the exploding database of genomic information, two types analysis techniques needed to be developed. Okay, two types of what? Two types of techniques needed to be developed. The first is bioinformatics, computer-based method to store, analyze, and compare genomic information. One of the bioinformatic tool is, MegaX software that can be used for checking the diversity or comparison between two pathogen to check their diversity. More are there. This is a generic capability with only minor differences between organisms. The second is functional genomics, the ability to experimentally test the function of genes and DNA sequence. The method used differ for each type of pathogen and plant host. Both sets of techniques can be said to be central to the skill set. Yeah, for a modern plant pathologist, the for this will be for this will be the information for the budding plant pathologist, is that please learn the bioinformatics and the functional genomics so that we can uh, work on the database of genetic information, because this is the future. Now what? The worldwide development of plant pathology as a profession. Plant pathology became recognized profession in the late 19th and 20th century. There were developments of research institutions, teaching programs, extension service across countries. Here, the first dedicated professional body of plant pathology was Netherlands Society of Plant Pathology. Now, here we have to remember that although there are several societies related to plant pathology, but among those three, we have to remember by heart. One is Netherlands Phytopathological Society, because it was the first plant phytopathological society, and also it is the oldest. Obviously, it is the first, it will be the oldest, right? The next is APS. Here I got it right. APS, that is American Phytopathological Society, which was established in 1908. Okay, Why to remember this? Because it has the largest, it is the largest society, means it has the maximum number of members. Okay, largest society. So Netherlands Phytopathological Society is the oldest and American Phytopathological Society is the largest. The next that we need to remember is IPS. What? Indian Phytopathological Society. Indian Phytopathological Society. It was developed by Mundakar in 1948 at New Delhi. Okay, and the first president was Dastur. Okay, because being an Indian, knowing about the greatest society of India, Phytopathological Society, it's very important, and lots of questions come from this section. Okay, I have overwritten it like on top of the text, but I hope you can see it through. Again, the oldest is Netherlands Phytopathological Society, the largest is APS, and Indian, that is IPS, Indian Phytopathological Society, established by Mundakar 1948, New Delhi, and first president was Dastur. Okay, so yeah. Rest, let's see, International Centers for Agricultural Research. In this, we just need to remember a few points like it was established to enhance global food security and reduce crop losses, especially in developing countries. It also focused on research, training, technology transfer in agriculture, also in plant pathology. One more thing, there is a term called CGIAR. All these international centers operate under the umbrella of CGIAR. The full form is Consultative Group on International Agricultural Resource. Where is it written? It will be written somewhere. We just need to search it. So what did I say? It is Consultative Group on International Agricultural Research. CGIAR. Okay? Okay, man. That's it for here. Only two more points are left that we need to discuss. One is the current status. Where did I left the current? Yeah, I got it. The current status of plant pathology. That was the history. So we should know what are the developments that we have done and what is the current status. Okay, crop protection relies on four principal strategies: biosecurity, agronomy, chemistry, genetics. So biosecurity refers, this is us, this biosecurity is us. So biosecurity refers to the efforts to maintain a physical separation between a pathogen population and a susceptible crop or natural ecosystem. This involves attempts to stop pathogens at national and regional borders and to eradicate invasive pathogens. That's it, this much, it's fine. Okay. So in future, for future, what we need to know, that is, the science of plant pathology has advanced substantially in the last 20 years. We now have a detailed understanding of how pathogens cause disease, how plants resist pathogens, how they develop resistance to pesticides, and how they evolve in the field. The last decades can be described as the era of model system in biology. So yeah, if plant pathology can develop so much in the last 20 years, then just imagine uh, with the developing technologies, with the developing infrastructure, involvement of genomics and artificial intelligence and whatnot. So what we can do in the coming 20 to 30 years. Okay. Okay. Let's see. There are many reasons to be optimistic about the future of plant pathology and our contribution to food security, global change mitigation, and biodiversity conservation. As long as communication routes are open and resources can be moved to where they are needed, the future is bright. Lastly, let's discuss about nomenclature. Nomenclature, naming. Directly, I'll come to this para, that is, a stable nomenclature is desirable as it reduces confusion and makes text searching more efficient. Okay, now we have so many countries, like 195 countries, and in India we have 28 or 29 states, right? So in different areas, each host, each plant, and every pathogen, they are named in their local and regional languages, so it's very difficult to come up with a strategy, a management strategy, and if it is prepared in a single country, in one country, so it will not be understandable in the other countries. So we have to come up to a stable nomenclature which will be understandable for in everywhere else. For that, we have this EPPO database. Okay, that is European and Mediterranean Plant Protection Organization, and now called EPO codes. So EPO codes consist of 5 or 6 consecutive capital letters derived from 4 letters of the genus name and 2 of the species. Crucially, the EPO code codes not change even if the species name does. In addition, the database lists the host names and the disease name in English and manual language. So here you can see example, go through it. Okay. That's it. We have come to an end. And as promised, I have attached the takeaway questions in the description box. Make sure to check them out. If anything still feels unclear, just drop your doubts in the comments. I will be happy to help. Thank you so much for watching. If you found this helpful, please like, share, and subscribe. And don't forget to hit the bell icon to stay connected with our PhytoPathoVerse family. See you in the next episode. Till then, keep learning, keep growing. See ya.